Good morning to everyone in the room and to those on Zoom. It's my pleasure to welcome you to the 126th meeting of the MDI Biological Laboratory Corporation. We have a full agenda today, including some important updates from Dr. Haller, and he will talk about the cutting-edge research that's going on here, as well as our plans for the future, which are actually quite exciting. So with that, I would like to call the business meeting to order. Can somebody make a motion to do so? Thank you. Seconds? Any opposed? Hopefully not. Okay. The first order of business are the meeting minutes from the 2023 annual meeting of the corporation. A copy of those minutes were provided electronically to everyone who registered for today's meeting, and hard copies are also available for those attending in person. Can I have a motion to approve the minutes from the 2023 annual meeting? Second? All in favor? Great. We will now turn to the committee reports, and the first one will be from Dr. Jan Coates, presenting the Secretary's report.
Thank you, Alan. I'm pleased to report that we have 199 members of the corporation. So very good and nice to see so many of you here. As that number breaks down, we have 6 full members, 5 associate members, 155 life full members, 33 life associate members. So that totals our 199. I'm also saddened to report that we have 3 members of the corporation who passed away last year. And they are full life member and former trustee, John H. Forrest, Jr., MD, Carl J. Carnegie, Jr., PhD, and full member, Mr. Thomas Rush III. And that concludes my report.
As is our custom, can we please observe a moment of silence for the members that have passed away? Okay, we're now going to turn to the nominating committee report, and that is Dr. Ed Benz. Can I have a motion to approve the Secretary's report? Thank you.
Thank you, Alan. Good morning, everyone. I'm pleased to offer the nominating committee report. The nominating committee recommends the following slate of nominees for re-election to the Board of Trustees for a 4-year term. That'll be the class of 2028. Jan Coates, PhD, Ruth Sayre, Dennis Schubert, MD, PhD. And we have a new member of the class of 28 that we propose for first appointment, Dr. Benjamin Humphreys, MD, PhD. Dr. Humphreys is a nephrologist, an esteemed internationally recognized nephrologist from Washington University, who has been on our scientific advisory board, involved with the lab for many years, very supportive. And we are delighted to recommend him. So could I have a motion to approve that slate? Second? All in favor? Any opposed? Thank you. We also are happy to propose the following slate of nominees for the 2024 nominating committee. That is myself as chair, Barbara Belts, PhD, Jerry Bowers, John Hayes, Wistar Morris III, Patricio Silva, MD, and Daphne Trotter, Esquire. Could I have a motion to approve? Second? All in favor? Any opposed? I also want to note with thanks that we had one person stepping down from the Board of Trustees this year, Ms. Ann Lehman, and we'd like to thank her for her service 3 terms from 2012 to 2024. And that's the report of the nominating committee. Thank you, Alan. Thank you all.
Boyd to give the treasurer's report.
I'll report first on the financial position. The financial position compares assets and liabilities. The positions at the end of 2023 are shown in the far right and are compared with positions of the prior year, 2022. First, as you can see in the first line of the table, the cash position of the laboratory amounted to $5.4 million at the end of 2023. This amount decreased during 2023, reflecting the use of over $2.5 million in reserve funds for strategic initiatives. As shown in the second line, receivables, including grants, this amount increased in 2023 by over $150,000 from 2022. Assets held in investment type accounts increased by $1 million. This is shown in the third line, investments, and this line includes the endowment as well as other things. This was mainly due to increases in the value of the securities held. The investment committee and the laboratory's outside investment advisor closely monitor the investment portfolio. As an update to these numbers, the market value of the endowment has increased over $450,000 as of the end of June 2024. And as of the end of June, the actual endowment per se amounts to $8.9 million. For liabilities, the main category is bonds payable. The institution continues to pay down the $5 million bond that helped finance the Morris Laboratory building in 2007. We make regular principal and interest payments. $3.19 million in debt remains to be paid off, and there's a schedule for doing this by the year 2036. The category called other liabilities increased by $102,000. This was influenced by general increases in accounts payable. Considering total assets and total liabilities, current assets continue to exceed liabilities by a significant amount. Net assets are positive and are shown in the red box. Net assets encompass assets with donor restrictions and assets without donor restrictions. The total net assets amounted to $23.2 million at year-end 2023, a decrease of $1.6 million from the prior year. The $23.2 million of net assets are distributed among 49 different categories or funds. The largest category is plant assets. This is shown in green, and this includes land and buildings. The more liquid general unrestricted funds and the reserve funds are shown in blue and red, respectively. The general unrestricted funds, the reserve funds, and the plant assets do not have restrictions. Other funds may be restricted by the donors for specific purposes. The institution may use all or a portion of the investment income that these restricted funds earn. Reserve funds decreased $2.32 million from prior year due to operations and the initiation of expenditures on the strategic plan initiatives. In addition to the funds listed above, the Maine Community Foundation manages the Salisbury Cove Research Fund, established by Tom Marin and the Marin Foundation. Funds are made available each year, and as in prior years, we heartily thank them for their ongoing support of the NDI Biological Laboratory. This slide shows the statement of activities. Activities include operating support and revenue and operating expenses. The numbers are broken out by those with or without donor restrictions. Total operating support and revenue in 2023 was $14.4 million. Total operating expenses were $16.8 million. This resulted in a net decrease of net assets of $2.4 million from the prior year due to operations. The non-operating net assets experienced a positive change of $831,000 in 2023. This was primarily driven by the large increase in the investment portfolio values. In summary, in 2023, total net assets from combined operating and non-operating activities decreased by $1.6 million during the year, but at year end, the laboratory continued to have a positive net asset balance of about $23.2 million. The slide breaks out the categories of our operating expenses. Total operating expenses were $16.8 million in 2023. If we exclude depreciation, which doesn't involve cash, the operating cash needs amounted to $15.4 million for the year. Of these cash expenses, 72% or $11.1 million was for the combined research and education programs at MDIBL. This also includes monetary distributions to other institutions throughout the state of Maine through the INBRI program. 22% of the expenses were for management in general. This is shown in purple. These are the costs necessary for the operation of the organization and its INBRI program, such as administration, accounting, budgeting, legal, insurance, safety, public relations, grant reporting, and compliance. 6% of the fundraising activities was for fundraising activities for the purpose of obtaining contributions to support the institution, and that's shown in green. This chart illustrates how the institution covered the $15.4 million in operating cash expenses for the year 2023. 49% came in the form of federal grants, contracts, and subawards, and that's the blue and red sectors together. Breaking that out, 34% of the funding went to the MDI Biological Laboratory, shown in the blue sector, and 15%, or over $2.2 million, was distributed to institutions that collaborate with our faculty and colleges and universities within the state of Maine through the INBRI program, and that's shown in the red sector. As in previous years, we highlight 2 federal grants from the National Institutes of Health that continue to provide major support to the institution. The INBRI grant brings in over $1.7 million to the institution and over $2.4 million in subawards annually. 2023 marked the 5th year of a 5-year award cycle, and under the leadership of Dr. Jim Kaufman, the MDI Biological Laboratory just received the next 5-year award. The INBRI program is currently in its 24th year. The other major federal grant to the laboratory is the COBRI grant. Under the leadership of Dr. Ian Drummond, the COBRI was just awarded its next 5-year annual award to enter into Phase 3. Phase 3 provides $1.25 million annually to support core service facilities for faculty research, such as the Animal Core, Data Science Core, and Microscopy Core. And finally, I want to mention that 2022 and 2023 were the initial years of a multi-year strategic operating plan at the laboratory. The strategic plan is designed to strengthen MDI's programs, its scientific excellence, its infrastructure, and its support, so that we can continue to achieve a critical mass of scientists and ensure long-term financial sustainability. The plan is being carefully implemented over time. In 2023, the laboratory continued to engage in controlled spending for growth while staying within board-designated guardrails to restrict spending for new initiatives, and the laboratory maintained the required cash balances needed for liquidity. Hang on. I wanted to make one more statement. The 2024 projections date, which we will review at the board meeting this afternoon, reflect a stronger cash position than anticipated due to major philanthropic support that aided the plan greatly in 2023 and is significantly bolstering our ability to continue to move forward toward our strategic goals. And we'd like to say thank you to the many supporters who have stepped forward. That concludes my report.
Thank you, Tom. Can we have a motion to approve the treasurer's report? Second? All in favor? Any opposed? All right, let's turn now to audit and risk, and I'd ask Daphne Trotter to present that.
Thank you, Alan, and good morning, everyone. This is the Audit and Risk Committee report. The MDI Biological Laboratory Audit and Risk Committee met with Julie Paquette and Olivia Fortier, auditors from the firm Baker, Newman and Noyes on Monday, June 24th, 2024. This was the lab's first year working with Baker, Newman and Noyes. With respect to their audit of MDIBL's financial statements for 2023, the auditors stated that MDIBL received an unqualified opinion with no material weaknesses noted. In other words, it was a clean audit. According to the auditor's written opinion dated July 1, 2024, the financial statements as of December 31st, 2023 represent MDIBL's financial position, changes to net assets and cash flows fairly in all material respects in accordance with accounting principles generally accepted in the United States. And that is the report.
Thank you, Daphne. Can I have a motion to approve the Audit and Risk Committee report? Second? All in favor? Any opposed? Okay, now to the good part of the meeting that you've been waiting for. And we are thrilled to have Dr. Haller with us, who is going to give us an update on what's going on at the lab and where we stand with the implementation of our strategic plan. So, Dr. Haller.
Thank you, Alan. I hope the microphone is working. Well, a little bit too high. Thank you very much for being here. It's always a great week. It's a big day for MDIBL. And I have named my talk The Next Chapter. Could I have the next? I can do this myself. Next slide. So what does the next chapter mean, and why the next chapter? I've worked at different institutions. Some of them were much older than the lab, this lab. Some of them were much younger. But I have not worked at any institution where the history of the institution is such an important part. And almost every day, we talk about the last 100 years and what happened during this time. And some of it is because we have written this all up. I don't know any other institution where we have 1,000 pages. Now, a new chapter, and it's called MDIBL, a Research Institute. And you may notice that a research institute, this is what we have become. We are not any longer a year-round institution, just mentioning, oh, and we are not a summer lab anymore. We are an institute. We are a research institute, a relatively small research institute in northeastern Maine. And I'd like to talk about these different parts: increasing our presence in Maine, campus development, faculty development, research programs, funding, translational research, education program, most importantly, science, but this will not be me, but Emily Spaulding will talk about that. And I try to be as brief as possible, which is very hard for me, but I try to be as brief as possible so that Emily has enough time to talk about her fascinating research. Now, increasing our presence in Maine, very important. Normally, I have this slide at the end of my presentation, and then everybody is half asleep already. But this was a very important part of our activities over the last couple of years, because originally, you all know this, famous researchers came here during the summer. We had a wonderful time. Everybody was dreaming about having a year-round summer, basically, which is not happening because year-round means now winter and fall and spring. And we were in Maine, and everybody knew that, but our connections were not that much and not that many.
as what you need when you're a Maine institution. This changed with the Maine IMBRI. And Jim Kaufman already mentioned. Jim, could you briefly talk about what IMBRI is all about? I know this is hard. 1 minute.
Thank you, Herman. I just want to say that, so the INBRI, as you heard, has been in existence for, 20 years now. It was started in 2004, and then its precursor, the BRIN, started in 2001 by Patricia Hand, who's here. And I think the INBRI, it's fair to say that it was crucial, instrumental in building MDIBL as a research institute. But it's not just about MDIBL, it's about the whole state of Maine. It's a network of institutions engaged collaboratively to develop the research infrastructure across the state by promoting cutting-edge research, not only of faculty at the different institutions, but at undergraduates who are working with those faculty. So I took over the INBRI in 2016 from Patricia, who had run it before then, and she showed me the ropes, was a fantastic mentor. We got it renewed in 2019, and we just again got it renewed in just this past May for another 5 years. And this map here, you can see all the institutions in the network. The whole point of the INBRI is to develop collaborations between researchers in Maine and connections, and to promote biomedical research excellence. I think we've done a great job, and as evidenced by the renewal, we're going to continue to grow that program. So yes, and on August 13th, we're going to have a celebration. Our Senator Susan Collins is going to be here, and some other congressional delegates, and we're going to have a big celebration about the renewal.
Because, and Jim is very modest, it's one of the best INBRIs in the country. The score Jim and all the others got was excellent.
Yeah, we got a great score, and we got funded, and we're very happy.
Thank you. So that's a wonderful basis. But over the last couple of years, we have added a lot of other institutions, and we closely collaborate. We have connections, as you have seen, with all the other research institutions, but COBE, we have student programs, we have a biotech program, and Andrea Tilden, who is Director of Education here, is also professor at COBE and very much still involved with their new developments. I have been teaching at COBE. We have an arts and science program with the museum there. So we are strong collaborators with COBE. Next one is the Rue Institute. You have heard a lot about the Rue Institute, founded 250 million 4 years ago in Portland. And we have not only joint appointments, actually they stole Jim Strickland from us, but Jim is still working at MDIBL. He still has a part-time contract here. So this has strengthened our relationships, and we have weekly exchange with programs. We have students here in my research group. I have a master's student from the Rue. We have 2 at the moment from the Rue Institute, and we have grants submitted. We have very strong relations with Maine Medical Center. So we actually have now submitted the first grant where we have patient materials from Portland, and we are exchanging research and research activities, and also with the Maine Medical Center. So there are regular meetings now. It's not just one project. And then next, MDI Hospital. They have a new education center, and we had meetings. We will give seminars here. We have me, but also Anastasia, and we will teach on rare diseases, on a kidney disease at MDI Hospital. The Bigelow Lab, you will hear about a fascinating project. So I knew that the Bigelow Lab was there 6 years ago, but now we go there on a regular basis. We have close associations, we exchange research projects. The University of Maine, obviously, we have students from there. We are part of their program. and we have joint appointments. Romain Madeleine is part of their COPRI, and an important part of their COPRI. And I am member of the Institute of Medicine there, and we have regular meetings. I'm on the board there. So we develop and we plan much more together than ever before. University of New England, we have research projects, and last not least, very important, the Jackson Lab. The Jackson Lab is changing. It's still a very strong research institute, but their other part, the mouse industry, is becoming more important. They have a new CEO, sorry. And but they also have a new research director. We have joint appointments. We have researchers working here and at the JACS with their own labs. We use core facilities together. We are still talking about a housing program. We have research programs in stem cells and kidney. We have regular meetings with the JACS every month, 45 young researchers sitting there discussing kidney. How's that for a small island? I can tell you I've never had this as a nephrologist over the last 40 years. So what this means is that we are now much stronger connected within Maine. We have regular meetings. MDIBL has become a research institute which is part of the Maine community, and they plan with us, and not only with the different institutions, but also on a governmental level. And sometimes they talk about MDIBL in Augusta without us even being there. Campus development. We have grown considerably. This is 78. This is about 10 days ago. It was terribly hot. This is 78 out of 94. And these are not our summer students. These are 94 employees of the lab. So that's quite an institute. That's a lot of responsibility, and it's a wonderful team. We all came together 50 minutes before we took this picture. Nobody was around. And then they all came from the different research labs and administration, and we met there. So as I said, it's a wonderful team. It's a great responsibility to keep us all together. But we also have to change our internal structure. Don't even look at the individual names, lots of people, but you can imagine that in order to make decisions here, this is not all going through the president, or more importantly, through Jerry Bowers, who is essential for that. We make now decisions together. We have an executive team where we meet regularly. We try to make decisions fast. We don't delay, so that everybody else can really work here, and we keep the institute going. This will change over time. We're still reorganizing. We're putting things together to make us more efficient. So you can see it's a wonderful institute, but when I remember and think back history 30 years ago, this was much more on the fly. It's still, but not that much as it was before. And then campus development, we put up the first new building in a long time, and this is student housing, the new student dorm. You can see this was the first picture taken, and now it feels like it has been there all the time. And this is what we aim for, that we renovate, we rebuild, but we'd like to keep the spirit, and we'd like to keep this special atmosphere of the campus alive. And also very new, we receive federal funding for the renovation of our research buildings. Not so much for the basic research aspect, but since we are translational, we are biomedical, and with a lot of support from Maine senators, we have received for the first time in 23 for the renovation of the Halsey lab. It still looks from the outside like old MDIBL, but it's very different on the inside. 1.6 million for the renovation, complete renovation. We'll lift the roof. We'll have the offices adequate for researchers which are taller than me working in there. And then at least we're in the budget. We don't have the money yet for the renovation, finally, of Bowen as an educational center. You're all aware that federal money is difficult to get. So first of all, we have to be in the budget, then the budget has to be approved. And once we have that, then we write a grant for the money, and the grants are complicated. So the process takes almost 2 years, but we are very optimistic that for 26, we'll have the opportunity perhaps to replace Karnofsky. Difficult because such a lot of history is there, with a new modular lab building. Very early, we even haven't talked about this with the board, and it's too early. But I think Jerry and I believe that we have a chance to apply for the money to get this new building. And this was all possible because of the support of the board. The new building you have just seen would not be there without the efforts of Dennis Schubert and Maggie Myers and the committee, and they were very supportive. It would not be there without our facilities. So our team, together with the board, has done a great job here. And now, most important, faculty development. Because the whole research institute is only here because of our research. Whoop. I can even go back. So this is the research at the lab at the moment. These are the 10 research groups, and I'll go briefly through that. As I said before, the research will be presented by Emily Spaulding. The Spaulding group only joined MDIBL, and we are very happy about this earlier this year. So these are the 10 research groups driving the research. You can see down here another 5 research groups. That's part of our strategic plan. That's what we are aiming for over the next couple of years. And 15, and the role model, as I mentioned before, often is Howard Hughes and Max Planck. That's the critical mass you need in order to make world-renowned science. Let me briefly use a slide Jerry Bowers has shown yesterday, just that you're aware how long it takes to build a research group. And to build a research group which is fundable. So what you see here, you have a hypothesis and preliminary data, experimental approach and verification of data. Then you write an initial publication. You have reviews, comments, and then you publish eventually. And when you have published, then you write a grant. So during this time period, this is what you get as a package. This is where the institution supports you. And this means other funding or philanthropy. And it also means that we have to recruit the most excellent, enthusiastic, a genius, cares for people, good manager. That's what a PI stands for. And to be honest, perhaps some of them have a training as a scientist. We hope so, and they're excellent. Not all of them have training as a manager. I was running a department in Germany with 135 people without any managing skills, and I had a budget of 21 million, which was my budget, and I was a medical doctor with a little bit of scientific background. So this is what a PI is. We need students. We need technicians, postdocs, visiting scientists. And this means a lot of effort, to put this all together. We have visiting scientists here. You see here under the management of Amy Richards in 2014, a little bit less than last year, but we have had this before. The most important change in order to achieve this are students. How do you bring students to MDIBL, not only for the summer, but long term? And we have done this over the last couple of years with your support. So we have now a graduate student program where graduate students from other institutions are coming to MDIBL for their whole project for 3 to 4 years. And these are all the graduate students working on campus at the moment. I have to admit, Anna left us because she graduated successfully 4 weeks ago, which is wonderful. And some of the others, like Sophia, she will graduate in beginning of August. So it's a lot of effort to bring them here. It's a lot of, how would I call that? Responsibility on our side to make them successful during this time. And it's a lot of effort. And Hannah Lust is doing this together with Andrea to keep this all together. So it's great to have them here. And I think we have quite a few of them here, and you're more than welcome to talk to them afterwards. A round of applause for the graduate students who are actually carrying the work. And as I mentioned before, this is the new building. We have now all the buildings we need to house them on campus. Our infrastructure has increased. You've heard that we had 2 years ago the first washing machine. We have now 3 washing machines on campus. We can provide transport for our students. How's that for a world-renowned research institute? And as I said before, this is because of our board supporting our strategic plan, but also, and you've heard this already when we talked about finances, wonderful supporters of the lab in these difficult times to get started and to get the institute started. So I'll go very briefly through the different research groups just to give you an impression. That's the Disney lab, that's the Kaufman lab, that's Dustin Updike's lab. And Dustin, you will be the only one. Where are you? Oh, over there. Dustin's lab, we have, you will see this during my presentation, we have more than one. Emily Spaulding, who will talk, is a graduate from his lab. And Liz Marney came back in education. She's a graduate from his lab, a well-funded lab. Dustin, at the moment, a role model. We already gave you this recognition last year of mentorship and taking this responsibility. This is Ian Drummond's lab. And here you can see that we are also changing. We are more diverse at MDIBL than we have ever been. The mix, and the researchers here know that this is very important to get grant support these days from NIH in the first place. So Ian, wonderful mentor, is doing a great job educating his research group. This is my research group. Actually, it's only part of it because I couldn't get them all together. At the moment, 2 are in California, and so, but nonetheless. A very nice example. Lots of discussions in the lab. Do we go back to work after such a sunrise hike? Yes, we do. This is the Godwin lab. Now, this seems to be small, but don't forget, James Godwin has a joint appointment. He has a research lab twice the size of this at the Jackson Laboratory. He's going back and forth between these 2 labs, and there will be more graduate students later in the year. This is the fastest growing research group. That's Prayak Muravala standing very modestly behind that tree. And this is the Madeleine lab, also fast growing. Romain told us yesterday evening, he came in 2021, and he is doing now great research with the group. This is Halina's group. This photograph was taken yesterday evening because she only arrived yesterday evening. So they took, and this is Emily Spaulding, and Emily, you'll talk about your research in a moment. So this is the faculty. And we are starting. You've seen that. It takes 4 or 5 years before you can actually lift off with a grant. And then you develop from then on. And Ian will talk this afternoon during the board meeting about more detailed strategies. Your wonderful slide with all these little blue figures. We'll talk about this in detail. I know this is a complicated slide. To be honest, when Cherry Bowers looked at my slide, she said, Dr. Haller, I'll lead you through. The message is relatively simple. So we have individual research grants, we have philanthropy, and we have collaborative grants. And as you can see here, these are the different labs, and these are real data. To obtain a grant is difficult. At the moment, you can see this. We have not an easy time beginning of 24. And then you get a grant, and then there are ups and downs. We need, for the individual careers and for the support of these research groups, these individual grants. But from the perspective of the institution, it's very difficult to run a budget based on that. So during these periods, this is especially when we need philanthropy. And we need philanthropy, and we've not distributed just like this. We have built a discovery fund, the Morris Discovery Fund, where our researchers, when they need support, apply to, and we want them to really think carefully about what they need and what they want to achieve with this money, and then we use philanthropy to bring us to this level. But even more importantly, we need collaborative grants, because only when you have a grant like the INBRI we just heard about, or Ian's COPRI, only when you have a grant which covers 5 years and the COPRI 10, then you can plan, then you can grow the faculty. So our plan is, and you can see here, this dip is because this has ended now. So we need a new COPRI, and we need more collaborative research projects, and we are working on that. I won't go into details, but Ian is leading developmental plasticity in regeneration. RMP biology will be the basis for our next COPRI, and Dustin Updike has taken over the responsibility. I mentioned the kidney group. The kidney is so strong at the moment, and we have so many different groups there. We'll talk about this afternoon, and especially tomorrow during our meeting with the Scientific Advisory Board, that we don't know what we will make out of that. And then we have citizen science and environmental health, and Jane and Jim will work on a collaborative grant. However, since the core of an individual research group is an NIH-funded R01, we have learned from our mistakes, and we have now criteria before submission. So we submitted too many grants without publishing beforehand because we couldn't wait. The research has to be significant, and we discuss this now more within the institution. We have regular presentations, which is obvious. We have a wonderful person, Stephen Sampson. Stephen, where are you? There. Stephen Sampson has been working at the Jackson Laboratory and has been writing their grants for 20 years. And now he's here, and we are very happy, and you're happy. And I know that we will write grants and publications together. We also have now more critical review of grant proposals by the scientific director and senior staff. Senior staff means that even non-scientific people at MDIBL can read and should read our grant applications, because if you're an intelligent person and you don't understand the science in the grant, something's wrong with the grant. So we'll use this as well. And then we have our scientific advisory board, and they are wonderful. They have told us, send us your specific aims, and we did. And they give us back reviews. Some of them are not very nice. I mean, I won't say names, but I've sent in my scientific, and I think they are wonderful. And then I get them back and they are totally changed and stripped to the bare bones. And this is what we need. So this is what we're doing at the moment. Perhaps we should have started this earlier. And where is Amy? Yeah. So Amy, she has to deal with all these grants. You know, writing a grant is relatively easy to hand it in, but we have made a promise to her, because it's so frustrating to hand in these grants and then tell us the negative results. We will only hand in the best grants ever in the future. And thank you very much for all the critical reviews and all the work you put into the development of our research and the faculty. Translational research. You may remember we talked about this a lot, and we still have the same aims, that we want to have research partnerships, tech transfer and commercialization, biosense education, and this is still very important. Literacy in biomedicine is something we want to contribute to workforce development within the state, and we are known for that. We are communicating this with other institutions. The first point here was critically reviewed by the Scientific Advisory Board, and they made strong recommendations. And we follow these, and we have learned the hard way that it's too early to form partnerships with pharmaceutical companies, because our animal models are still not there. They are all still using the mice. But nonetheless, tech transfer and commercialization and using our animal models, I want to present two projects briefly. So our collaboration with the Bigelow Lab. That's a wonderful project, and it's within Maine. So the Bigelow Lab is a national center for marine algae. They have 3,000 strains, and they make extracts of these algae in order to find out whether there are substances in these algae which are useful for new medication. And this is a collaboration with Northeastern. So they have this lab called Battle, which uses structural analysis and mass spec to identify the compounds which are in there on a structural basis. And then they can model this. They have made compound identification, and they can synthesize new compounds. So they receive these extracts, and we receive them. And Jim Kaufman has started to work on that. We are working on that. We're using our animal models. So here we have a fish with inflammation. We have a fish with complement activation. We have a fish with kidney injury. And we have started now to look into other animal models. And we test these compounds, these extracts, and see whether we have a biological effect. And then the data, we do gene arrays. We try to identify target, and they try to identify compounds. And then this goes to the Roux Institute, and they do the very complicated modeling. When we have a target and they have a structure, does this fit? And how we can develop this further. And then we go back into the different labs. And this is not only a dream. We have started pilot screening. Jim has the first positive result. And the initial focus is on inflammation, and we have already submitted one grant. And NSF has said, well, that's a good project. You have to rework the application, but we may fund the first steps of that. And then patents, translational research. And we have a couple of compounds or methods where we look for feasibility, but we have one wonderful patent, and I'll show you a slide here. So this is ML233, and it tells you a story. 10 years ago, this slide would have filled half of my presentation of MDIBL. Because where we are here is we have a new compound discovered in the Medellin lab. So this molecule inhibits melanogenesis, and you can use it as a cosmetic skin product or as therapy for pigmentation disorders, for melanoma therapy, adjuvant for metal-based chemotherapy agents. We have, you can see this here, a preliminary patent, and we'll have the full patent in a couple of weeks. And not only that, but we already have, we're talking to an entrepreneur, and this person had already had a look for 4 weeks at the compound, seriously interested. So now we're talking about a license. The lawyers are looking into that, and we'll present it to the board and discuss this. And if this goes according to process, we'll have an STTR grant application in September. So that's already there. It's not only a patent, it's not only a discovery, but it's already in the arena where it could become translational. Last but not least, our educational program. This is research training and education for lifelong learning. And we have 4 parts of that. K-12, SEPA, this is boot camp for high school students, outreach for Maine community program, family science night, other internships. And we are very proud that Liz Marnick, Liz, where are you? Yeah, Liz Marnick has joined us, and she is another graduate from Dustin's lab, and she has worked at Hassan University, has been very successful there. And now we're very happy that you're back here, and this is the whole program you're going to run. Our undergraduate program, the graduate students and postdocs, and we have talked about graduate students, and hopefully we can talk about, not next year, but in 2 years, about a successful postdoctoral program financed externally. And then last but not least, Chris Smith with courses and conferences. And you have to look at the program this summer. We have wonderful conferences. We're also focusing with our conferences to have international, national conferences of important for microscopy, for stem cell, and so the topics of our research have to be communicated by our courses and conferences. And I'm almost there. Our latest small child is arts and science. And you have seen this before. To be honest, I should have included our newest toy. Where is Priya and Marco? Yesterday evening, I have to admit, first I was very critical about these things. I thought, you know, this is just a toy, but it's wonderful new science. It's a totally new way to look in 3D terms and to measure things. And I feel like one of these gentlemen I know from the 17th century looking through the microscope for the first time and seeing new things. So you should all have tours with that, because you can really fly into the tissue. You can identify in 3D where you are, and you can measure it immediately. So this is wonderful. We have small exhibitions, and you're more than welcome to go on a tour on images of science, making the secrets of nature visible, just to make it more transparent on which levels do we actually generate pictures, how do we look at nature. And this is an MCBI, and Emily Burke has done a wonderful job in setting this exhibition up. We have seminars and lecture series. Most importantly, we collaborate with other institutions on MDI, the Historical Society. And for next year, we are planning Science in the Garden together with the Garden Society. But also with Kobe College, we had podiums discussions about art and science in their museum. And we have, what I like a lot, these museums tours. And let me finish with that. This is us with our donors. Thank you very much. And this is in DC and this is in Philadelphia. Thank you very much, the Morris family, for organizing this. And this is Dr. Lavoisier. This is in the Met, and he is here with his wife. And you can see, very modern. The wife is more important, looks much more important than he does. Unfortunately, he went to the guillotine 3 years after that, not because of the science, but because of financial implications. With that, I'd like to thank you all, not only for coming, but for helping and supporting this wonderful institute. And I'd like to finish, if I can. Can we go back? One more. 2 remarks, and some of you have seen this before, but I like these quotes so very much. Program director, NIH, 2 years ago, all the great minds are moving to MDIBL these days. There must be something in the air up in Maine that allows for free thinking, creativity. Yes. And then, and we can really use this. I have asked permission for that. We consider MDIBL a national treasure. That's what the NIH thinks of us. Perhaps not so much in terms of research, but in terms of tradition. We'd like to keep this. And last not least, this was coined by Ian, MDIBL, the way science should be. Thank you very much for your attention. Can we go to the next talk? Because this is now the real thing. So Emily Spaulding is from Maine. And she graduated from the University of Maine. And then she did research both at the Jackson Lab and at MDIBL. And she was very successful. She worked on neuronal disease at the Jacks. And she will bring this expertise to MDIBL. So all of a sudden, neurology, and I'm fascinated by this, and chronic neurological disease will be important. She published very well. And we are very lucky that she did not choose to go to the Jackson Lab. You could have done that, where your first science publication was from. But she chose to come to MDIBL. She graduated from Dustin's lab with a very nice publication in Nature Communications, and you received a wonderful award from the NIH, and the program director responsible for us, John Lord, actually had it in his calendar that he had to walk over to the other institute and shake your hand, because it's not only us being proud of you, but NIH is. And you will give us insight into what you're doing. I have to mention this because she is not only a wonderful researcher, but also a role model. I wouldn't call it life-work balance, because I don't know how you balance this out, but you have 4 children and you are doing this together with your husband and a wonderful family. And I've met the children very briefly on our parking lot recently. You will not talk that much about that, but about your science, and it's all yours. And to be honest, you have 35 minutes, 34 minutes.
You don't have to use all of that.
Thank you. All right, microphone on? Yeah. Well, thank you for that introduction. I'm so pleased to have landed here at MDIBL for this next phase of my career. And actually, my very first graduate rotation was in 2013 here at MDIBL in the same exact lab space that now houses my research group. So things have really come full circle. So I'm going to give you a little snapshot of work that I have mainly done as a postdoc with Dustin Updike, and am now bringing with me as I transition to my own independent research group. And in my brand new lab, we are fascinated by the cell biology of this beautiful cell type, the neuron, and how its unique cell biology could underlie development of neurodegenerative disease. So going forward, the big overarching question that my lab hopes to address is what drives neurodegenerative disease. And the forms of disease that I'm referring to are age-related progressive forms of disease. Typically, symptoms don't manifest until the 6th or 7th decade of life. But once degeneration begins in mid to end of life, there's really very few strategies available to slow degeneration and virtually no therapeutic strategies to stop regeneration or reverse its devastating effects. And ultimately, patients succumb to very devastating symptoms and death. Those variety of symptoms will be different depending on the exact disease diagnosis, and that largely underlies the fact that each one of these diseases primarily targets a different neuron type in the nervous system. For example, ALS is a motor neuron disease, which targets the upper motor neurons in the motor cortex and the lower motor neurons of the spinal cord. So this is what underlies the loss of motor control in these patients. But despite the wide variety of neuron types targeted in these age-related forms of neurodegenerative disease, there are thought to be common disease mechanisms. One such mechanism is disrupted cell organization. And we know that neurons are especially reliant on proper cell organization because of their highly polarized morphology. For example, a human adult motor neuron can extend an axon up to 1 meter away from its cell body. So how does the cell maintain precise organization over such vast distances? Well, neurons have a few different tools in their toolkit. One common mode of organization that we're all familiar with is the use of membrane-bound organelles, such as the ER and the mitochondria that sequester all of the molecules needed for a given biological reaction by housing them in a structure enclosed by a membrane. But just as often, cells use an alternative mode of organization that doesn't require the use of a membrane and occurs when functionally related biomolecules condense into liquid droplet-like structures called non-membrane-bound organelles. These non-membrane-bound organelles are very dynamic so that cells look a lot less like this and much more like this, where these liquid droplet-like structures are constantly moving, fusing, bouncing off one another, and they can rapidly assemble and disassemble based on changing cell conditions. There are dozens of examples of non-membrane-bound organelles in just about all different types of cells, not just neurons. A few types that I'd like to call out are the nucleolus, which is inside the nucleus. The nucleolus is the largest non-membrane-bound organelle inside cells, and it houses thousands of proteins and RNAs that are mostly there to help with the process of ribosome biogenesis. In the cytoplasm, stress granules function in translational control, very important for neurons. RNP granules are extremely important for neurons. They function in trafficking molecules like mRNAs down the long axon for use in the axon terminal. And then neurons even have some unique forms of non-membrane bound organelles that are only found in this cell type, including synaptic vesicle condensates and the active zone, which is a liquid-like structure that organizes all of the machinery needed for neurotransmitter release. So disruption of each and every one of those types of non-membrane bound organelles that I just called out by name is strongly linked to neurodegenerative disease. In fact, some of the most well-known proteins, the major players in age-related neurodegenerative disease, are known to form these liquid droplet-like structures in healthy neurons. But in neurons of individuals with neurodegenerative disease, instead, these proteins are misorganized and they form these clumped aggregate-like structures. So the question is, how does the misorganization of these proteins drive neurodegenerative disease? And we really need to study this question inside a living animal because of the dynamic nature of this form of cell organization that I just illustrated to you with this video. But in mammals, neurons are fairly inaccessible. They're typically buried deep inside the body. So to overcome this challenge, our lab is using the model organism C. elegans, which is uniquely suited for the study of this dynamic form of cell organization inside a living animal. First and foremost, they're optically clear, so they're highly amenable to live imaging experiments. In this picture of an adult C. elegans hermaphrodite, we can easily pick out some of the major regions of the body, including the pharynx and the head, the intestine, and the germline. C. elegans are also highly amenable to CRISPR-Cas9 genetic engineering. And so, for example, we can inject all of the reagents needed for CRISPR genetic editing directly into the germline of the animal because we can see it. And then we can screen for our desired genetic edit in the progeny of that worm. So by combining these two advantages, for example, we could use CRISPR to fluorescently tag a gene and then image the protein's dynamics inside a living worm. One other advantage of C. elegans I want to mention that's specifically relevant to our study is the abundance of knowledge that's available for the C. elegans nervous system. It's known that each and every adult C. elegans hermaphrodite contains exactly 302 neurons and 56 glial cells. Their developmental lineage is completely mapped. Their morphology is well characterized, and their synaptic inputs and outputs have largely been identified. And moreover, over the last few years, some of the big C. elegans neuroscience labs have collaborated to provide the community complete gene expression maps for each and every neuron class in C. elegans. So with all of these advantages in mind, our strategy is to perform CRISPR genetic editing of neurodegenerative disease-associated genes, and then to perform super resolution live imaging of individual neurons in living worms. And I should say that super resolution imaging is one of the amazing techniques that we have available to us here at the MDIBL light microscopy facility. Okay, so here's where the story actually begins. Just about 2 years ago, Dustin and I published our discovery of the C. elegans homologue of the human protein nucleolin. Nucleolin is a highly multifunctional RNA binding domain that's most abundant inside the nucleolus. And if you remember, I mentioned that the nucleolus is the largest example of a non-membrane bound organelle inside cells. We named the C. elegans homologue nucleol-1. And nucleol-1 contains all of the same 3 protein domains that are found in the human protein, including a coiled acidic stretch, classic RNA recognition motifs, and an intrinsically disordered arginine glycine or Rg repeat domain. The very first thing we did when we discovered nucleol-1 was to do a CRISPR experiment. We fluorescently tagged endogenous nucleol-1 with a split GFP system so that we could exclusively visualize nucleol-1 in the germline. So why the germline? We chose this tissue because it's known that germ cells contain some of the largest nucleoli in C. elegans. So we knew this would provide us with optimal imaging conditions with which to first work out how nucleol-1 is organized into the nucleolus. Here's the results of this CRISPR experiment. You're looking at the germline loop of an adult living C. elegans, where we have germ cells that are maturing into oocytes as they turn the bend of the germline. Each one of these green spheres represents nucleol-1 inside the nucleolus of an individual germ cell. Next, we wanted to perform super resolution imaging of nucleoli in order to better understand the precise organization of nucleol-1 within this non-membrane bound organelle. So for this experiment, we tagged a second protein called fibrillarin or Fib1 with another fluorescent molecule, worm scarlet. And so in this still image, you're looking at 2 individual germ cell nucleoli. And then in the video I'm about to play, we've taken 1 single nucleolus and made a 3D reconstruction so that you can get a good idea of how these non-membrane bound organelles are organized in multiple dimensions. And so you can just kind of watch that video as I describe what we found. We found that C. elegans nucleoli are comprised of 2 immiscible separate subcompartments. Again, without the use of a membrane, which is pretty amazing. The innermost compartment, labeled in pink, is where this protein Fib1 resides among many other proteins. We call this inner pink compartment the fibrillar center, or FC, and we think that this is where the beginning stages of ribosome biogenesis takes place. You can see in 3 dimensions that this inner compartment forms a net-like structure. And then the second compartment, called the granular component, or GC, is labeled in green. This is where nucleol 1 resides, and this component surrounds and fills in that inner compartment. And we think that in the GC, this is where later stages of ribosome biogenesis occurs. So interesting to our study of neurodegenerative disease, it's been shown that disruption of nucleolin organization in the nucleolus is linked to the most common genetic form of ALS. So this was a paper published in 2014, and I'll just show you one panel from this paper. What they did is took human-induced pluripotent stem cells from healthy control individuals and from an ALS patient, and they differentiated those stem cells into neurons. So in the top panel, you can see a healthy control neuron. The cytoplasm is labeled in red, the nucleus in the blue, and nucleolin is labeled in green. And you'll notice that nucleolin is precisely organized into these 3 nucleolar regions. In contrast, in the ALS neuron, we see that nucleolin loses that precise nucleolar organization and is instead more dispersed throughout regions of the nucleus. But if and how nucleolin misorganization contributes to ALS pathology is unknown. This was done in cells in a dish. So now that we have identified a homologue of nucleolin in an animal that's conducive to live imaging and CRISPR genetic editing, we can start to ask the question, how does disrupted nucleolin organization contribute to neurodegenerative disease? So to do this, the first thing we needed to work out was what domains of the nucleol 1 protein might be regulating its precise organization within the nucleolus. And we had a hypothesis. We hypothesized that the intrinsically disordered arginine-glycine repeat domain, which is found in nucleol 1 and in the human protein nucleolin, would be in some way, shape, or form regulating the organization of this protein. And I won't go into all the details of that support that hypothesis, but just suffice it to say that at the time we did these experiments, there was a lot of in vitro data, so in a test tube or cells in a dish, that supported the role of these types of domains in organization of proteins into non-membrane bound organelles. But at the time, there was no data from a living animal. So we hypothesized that this domain would be important for nucleol 1 organization. To test that, we deleted the entire RG repeat domain from endogenous nucleol 1 in the strain in which nucleol 1 was tagged with GFP. And then we again performed super resolution imaging of germ cell nucleoli. In wild type nucleoli, you can see what I just described on the previous slide. The inner FC labeled with Fib1 worm scarlet. Here you're looking at 4 different nucleoli. And then nucleol 1 labeled with GFP is in that outer GC region. And together we have this beautiful subcompartmentalization of these 2 regions of the nucleolus. In contrast, when we delete the RG repeat domain, we see that both Fib1 and nucleolin can still get into the nucleolus, but they can no longer enrich in the correct subdomain. And instead, we have a general mixing of those FC and GC compartments. So this experiment was important for 2 reasons. The first is that it really gave us an example from a living animal that these domains are important for the organization of proteins into non-membrane bound organelles. The second important thing is that now we have a really precise model in which to test how disrupted nucleolin organization could contribute to neuron dysfunction. Because we haven't taken nucleol 1 out of the nucleolus, it's still there. The only thing we've done is precisely disrupted its organization. So now we can ask the question, how does disrupted nucleol 1 organization impact neuron function? And I have to tell you, I cannot answer this question for you today. This is going to take several more years of work, I'm sure, but I can at least tell you about a couple of experiments that we've done to start trying to dig into this question. The first thing that we did, because of the link between nucleolin and ALS, which is a motor neuron disease, we wanted to assess locomotion and movement in our worms as a proxy for motor neuron function. So for this, we collaborated with a company called In Vivo Biosystems, and they took 10-minute long videos of 30 worms per genotype, and they analyzed the movement of the worms in those 10-minute videos for 26 different features of movement. And what they found was really exciting. So let me just tell you the 4 genotypes that we included in this experiment. We have 2 wild type or control genotypes. In gray, just a plain old wild type nucle-1 worm, and then in green, a wild type nucle-1 with the GFP tag. This was important to include because we want to make sure that the fluorescent tag itself is not resulting in motor dysfunction. We also included another control in a way, which was deletion of the entire nucle-1 gene in blue, and then in orange, we have deletion of just the RG repeat. And what I hope you can find is what you can see is that we found a very specific motor dysfunction in the nucle-1 RG repeat domains, which manifests in increased motor activity and increased body wave initiation rate. In other words, they're crawling around more frequently on the plate. And so the results of this experiment are important for 2 reasons. First of all, this suggests that disrupting nucle-1 organization alone with deletion of the RG repeat is sufficient to cause motor dysfunction, suggesting that in human ALS patients, maybe that nucleolin misorganization is actively contributing to disease pathology. Now, the second point I want to make is, at first I was a little puzzled, and you might be too, by the direction of change in motor activity. I sort of expected maybe the worms would move around less because ultimately movement is inhibited in ALS patients. But instead, we saw a motor hyperactivity. And so I dug into the ALS literature a little bit more, and what I found was really interesting. So pre-symptomatic ALS patients commonly have hyperexcitability in the motor cortex that manifests in muscle spasms and twitches long before they show symptoms of motor neuron degeneration. And this hyperexcitability in the cortex is thought to be explained by an imbalance in excitatory and inhibitory neurons of the cortex. Likewise, the C. elegans motor circuit, although admittedly far less complex than the human motor circuit, is comprised of both inhibitory, in gray here, and excitatory motor neurons in white. And it's really the balance of these excitatory inhibitory motor neurons that controls the forward and backward locomotion of the worm. So I think with our result, we may have a really good model for the pre-symptomatic stage of a motor neuron disease. And so this experiment has allowed us to make the hypothesis that disruption of nucle-1 organization causes dysfunction of neurons of the motor circuit. And it's going to take some more experiments to figure out exactly which neurons might be involved in causing this motor hyperactivity phenotype. But to dig into this hypothesis a little bit more and kind of shore it up a little bit more, we did one more experiment. We decided to assay gene expression in our nucle-1 worms. So for this, Noah Lind, a research assistant in Dustin's lab, did a lot of this experiment with me. He synchronized thousands of young adult worms of 3 genotypes: wild type nucle-1 with just the RG repeat domain deleted, and then the entire nucle-1 gene deleted. And we did 4 replicates of each of these conditions and then performed whole worm mRNA sequencing. And for the sake of time, I'm just going to talk about genes that were downregulated significantly in nucle-1 RG deletion worms compared to wild type. What we found is that many of the most significantly downregulated genes in nucle-1 RG deletion worms were found in neurons. They were neuronal genes. And remember, this was sequencing of the whole worm. So you can consider this to be kind of like a blazing signal. And in fact, many of these genes are enriched in specific compartments of the neuron, like in the axon or dendrite and at the synapse. And when we look at the identity of these genes, we found that 9 of the most highly downregulated genes are core components of the neuronal synapse. Here's the neuronal synapse. This is a complicated diagram, but it contains a lot of machinery that is required for the release of neurotransmitter. This is how the neuron communicates with other cell types and with other neurons. So this experiment has led us to refine our hypothesis. Now we think that disrupted nucleal 1 organization might specifically impair synaptic structure and function in neurons of the C. elegans motor circuit. So over the next couple of years, I have lots of experiments planned for my own lab, and I'm just going to tell you about one main approach to sum things up that we're hoping to use to really help us to test this hypothesis, to help us look at nucleal 1 organization in neurons and to assess how synaptic structure might be changing when we impair nucleal 1 organization. And so to do this, we're making use of that resource that I mentioned at the beginning of the talk, where the C. elegans community has access to complete gene expression profiles for all 118 classes of neurons in C. elegans. This database is called the C. elegans Neuronal Gene Expression Map and Network, or CENGEN. And what we can do is query this database for genes that express highly and exclusively in one neuron subtype at a time. And then we can make a heat map of all of the genes that we've chosen and look at how they're expressed in different classes of neurons versus non-neuronal cell types. So for example, this gene at the top here, we would consider to be what's called a pan-neuronal gene. It seems to be expressed in all neuron types, but not so much in non-neuronal subtypes. So I'll just give you 2 examples of genes that we've chosen to express a fluorescent reporter system in 2 different neuron classes. The first gene is called TTR39, and I hope you can tell from this heat map, it expresses pretty highly and exclusively in this class of motor neuron called the VDDD motor neurons. These are inhibitory motor neurons. These neurons have their cell bodies on one side of the worm and project their axons on the other side of the worm, so they can successively relax muscles on one side of the worm or another as the worm performs that sinusoidal crawling motion. And so directly behind this TTR39 gene, we place sort of a complicated cassette. So I didn't include it in here, but it's a reporter cassette that's going to allow for the entire neuron to light up pink with a worm scarlet fluorescent protein. And then it's going to allow nucleal 1 to light up green. So we can observe nucleal 1 in individual neurons of any subtype that we want. So what does this line look like? I'm very proud of this line. Summer student in my lab, Ella Wouchet, I think she's here. She's back there. Her summer project was to fluorescently tag nucleal 1 in a line in which we already had pink motor neurons. And so I'll share with you, she was successful in that, and now she's well on her way to deleting the RG repeat domain from these worms so we can start testing organization in neuronal nucleoli. So what you can see here in pink are these VD motor neurons along this side of the worm. They're projecting their axons to the other side of the body. And here are the cell bodies. So nucleal 1 is labeled in green, but when it overlays with pink, it appears white. All these little white dots, I've pointed out some with arrows, are nucleal 1 inside the nucleolus of these cells. And same in this image. And so we've done a little bit of preliminary imaging of these worms. And of course, I had to do super resolution imaging to really see what the neuronal nucleolus looked like. And I don't know if you caught this from the germ cell nucleoli, but they're pretty big. They're like almost 3 microns in size. And these neuronal nucleoli are about 6 times smaller. They're about 0.5 micron. So I wasn't sure if we could, and you can't see this so well on the screen, but I wasn't sure if we could see any real organization or subnucleolar patterning in these nucleoli. And again, this screen is kind of messing up my big result, but you can see that in the middle of this nucleolus, there's a little bit of an empty space, as if there's a spot for that inner FC region. If we were to just tag Fib1 in this line, I think we'd see some subnucleolar patterning, just like we see in germ cells. And then my final data slide, I want to tell you one other line that has proved to be extremely beautiful. This is an uncharacterized gene that from SunGen we found expresses in these mechanosensory neurons. These mechanosensory neurons are not strictly part of the motor circuit, although mechanosensation is important in regulating locomotion. But we chose them because these neurons, there's only 4 of them, 2 in the head, and then 2 in the body, a left and a right for each pair. They extend these elaborate dendritic arbors that blanket the entire head or entire body of the worm and provide mechanosensation for the whole worm, just 4 cells. And so we thought with this very elaborate dendritic morphology, maybe cell organization is going to be especially important for this cell type. So that's why we're going forward with this line. This is a fancy video that Frederick Bonnet made. And what you can see are these 2 mechanosensory neurons in the head. The 2 dots are the cell bodies. And here are these elaborate dendritic arbors that cover the entire head. And then he makes a 3D reconstruction, and we can really get a good sense of the volume, the size of these dendritic arbors. And it's just really, really beautiful. I think will be an interesting line to study more. And then this summer, we also went a step further and tagged nucleol-1 in these cell types. So here in white again, we have nucleol-1 in the nucleolus of these cell bodies in the head, and then in the neuron type in the body. And when we perform super resolution imaging, we zoom in on this nucleolus right here. Hopefully you can see this a little bit better. I think we see some subnucleolar patterning. Right here we've got this little empty region that might house the fibrillar center. So I think that these lines are going to allow us to visualize nucleol-1 in these specific neuron subtypes that are relevant to our motor hyperactivity phenotype. And when we look at them in RG deletion worms, we'll be able to understand how organization is disrupted in neurons and hopefully ultimately link that to disrupted function. And one other just tidbit that I don't have time to talk about, but if you come to our poster session later today or tomorrow, this toolkit that we're using to express these pink and green fluorescent molecules in the neuron is very flexible so that we can actually kill the pink fluorescence in the entire neuron and instead put it on one of those dysregulated synaptic structure genes that we found to be downregulated in our worms. And so we can simultaneously look at nucleol-1 and synaptic structure in the same individual neuron. So that's kind of where we're going for the future. So in summary, I hope that I impressed you with the use of C. elegans for a wonderful model to study this dynamic form of cell organization in a living animal. I think our data is suggestive that disrupted nucleolin organization could actively contribute to ALS disease pathology. I think that our green and pink split fluorescent system is really going to allow us to dig more into how nucleol-1 is organized in neurons and how its disruption might lead to synaptic dysfunction. And then ultimately, several years from now, I hope that we'll be able to show a role for nucleol-1 organization in synaptic biology that will really give us an idea of how nucleolin could be causing neuronal dysfunction in ALS patients. Oh, can we go back one more so I can thank? Oh, it's frozen. Well, oh gosh. We'll get there. Well, I know the people, so I can thank them without their picture up there. I want to thank my tiny new lab that consists of summer student Ella, who I already mentioned, who's done a wonderful job making new strains this summer, and also my research assistant, Leo. Leo was actually a College of the Atlantic intern in Dustin's lab in 2021 and made the nucleol-1 deletion line. And now they've joined my lab as my research assistant, and I'm so lucky to have them. If you could have seen the state of my lab space a month ago with boxes piled everywhere, it was horrible. And they have single-handedly organized and set up my entire lab space, so we're ready to do science. And I also want to thank, of course, Dustin's lab and Dustin, who Herman was spot on. Dustin is an amazing mentor. He was for my postdoc, and he continues to be as I transition to faculty. And all of the members of his lab, Catherine, Noah, Hugo, Camila, other interns that have directly helped with experiments that I shared today and lots of experiments that I didn't have time to share. And thank you. We have time for, do we have time for any questions or am I done?
Yeah, I think there is maybe one or two questions.
Okay. One or two questions? Yes.
Can you explain why Parkinson's was not in that list?
Parkinson's was in the list on the first slide. Yes, it was. Yeah, so that's also one of these age-related forms of neurodegeneration that has disrupted cell organ.
In the words, human neuroplasticity.
In what regard? In terms of motion or speed. So the worms have a pretty classic movement, sinusoidal movement, which is really highly quantifiable. And it certainly changes with age, if that's what you're asking. It can be modified, yes. So we have, and yes, so when we make our precise edit in nucleolin, we see that there's a change in the worm's locomotion. Yes, so it can be modified with disease mutations. It speeds up, yes. It goes faster, so there's a little bit of hope, I think, yeah, yeah. Yes.
I suspect that multiple sclerosis could also be tackled through this kind of thinking too.
Yeah, so multiple sclerosis is a little bit of a different type of neurodegenerative disease. It hasn't been as specifically targeted to be linked to disrupted cell organization like these age-related class of neurodegenerative diseases, but I think what we're learning about these dynamic cell structures is that they're important for almost everything, so I wouldn't be surprised if we could throw that in there.
And the signal, the passage of the signals and
Exactly.
different parts of the cell.
Exactly, right. I'm sure these liquid-like structures are important for basic neuron function. So there's probably disruption of these structures in one way or another when you have neuron dysfunction in multiple sclerosis. Yes, thank you.
Stan.
One of the things that Dr. Haller didn't mention is that in the midst of all of Emily's amazing accomplishments, she was also a science teacher. And I'm always so jealous when I hear her talk that I didn't get a science teacher like that, because I just think it's amazing. And your work is so exciting, beautifully presented. Thank you so much. So good morning, everyone. Now we all need a little bit of a coffee break, right? But the good news is my presentation is quite brief this morning, and my message is really simple, and that is one of just immense gratitude, because I think you can see, based on Dr. Haller's remarks this morning, but also on Emily's, just how exciting the work is that's happening here and how much we depend on the amazing support that you all provide. So we are incredibly grateful for that this morning. Let's see if I can actually make this work. So earlier this year, as Dr. Haller alluded to in his remarks, we were in Washington, D.C., watching our beloved colleague, Patricia Hand, receive an award for her work with the INBRI program. So just recognizing the tremendous dedication and commitment she has to supporting and expanding research in Maine. And this quote came up that Dr. Haller mentioned. Dr. Lorsch said, gosh, you guys are 125 years old. MDIBL is really a national treasure that should be supported. So if you think this is the last time you've seen this quote, you are wrong. We are going to get some mileage out of that. I said, can I quote you? And he said, I hope you will. So it's a wonderful thing. But to those of us who are here, MDIBL, certainly our scientific legacy does kind of make it a national treasure. Our commitment to comparative biology, as you can see from the work that Emily presented, is something to be immensely proud of and something that the lab is recognized for internationally. But for those of us here locally, I think MDIBL is a little bit more of sort of the crown jewel of our community, right? That's a big part of who the lab is, how we relate to the lab. It's a gathering place. It's a place that brings people together, that unites us in a common purpose. And it also grounds us in this beautiful place that we are so lucky to call home. And so today, what I want to do is just talk to you a little bit about this moment in the lab's history where we are, which is really at a very transformative phase in our growth. So way, way back 25 years ago, when there was just a few of us, Claudine, you must have taken this picture because you're the only one that's not in it. There was all of 10 of us, I think, at the time. And then you can see the migration, 2006. I think we were somewhere in the neighborhood of 30 or so. And now here we are today. And so all of that growth has really been made possible by the tremendous support that this group has provided to the lab. And we are incredibly grateful. And I think one of the things that you certainly get a sense of, I think, in Dr. Haller's remarks, is that it is a very intentional growth, right? It's a strategic initiative to try and make sure that we maximize the opportunity, that we provide young scientists with an opportunity to explore these new ideas, that we utilize technology in a way that really enhances our ability to make a meaningful impact in the quality of the human condition. That's really what our purpose is here. But nonetheless, we also want to be thinking very carefully as we grow about maintaining those core values, right? The value of community, because I think that is what unites us. It is what really provides us with that kind of touchstone of being part of this group. And it is, in some ways, what sets us apart. So as we grow, you know, Dr. Haller mentioned we're no longer a seasonal institute. We've coined a new phrase at MDIBL. It's called Febuli. And that is really because there's more activity happening now during the off season almost than there is during the summer. And so I just want to give a huge shout out to our president's cabinet members, to our board of trustees for the amazing effort that they have put out there in terms of expanding, as Dr. Haller was saying, not only our presence in Maine, but really along the eastern seaboard as we begin to really engage this amazing community in moving the lab forward. So some examples, you know, that Dr. Haller shared with you, we are all over the place these days. We're in DC, we're in Philadelphia, really just working to reach out and continue to stay connected to this amazing community. So as you have heard me talk about before, as you've heard Dr. Haller talk about, we are in the midst of a $45 million strategic plan. And I want to give you a brief update today in terms of where we are in funding that strategic plan and how that is going. I think he touched on that just a little bit. But I want to give you an update. When I spoke to the corporation last year, I think we had raised about $14.5 million. And so I am very happy to say that we're making a lot of progress. We have raised $27.5 million toward our goal of $45 million. And I also want to take just a moment to recognize our board of trustees and the leadership that they have provided in making this possible. The trustees themselves have committed $20 million collectively to this effort. And you know, often I think they are unrecognized for the tremendous support and leadership that they provide to this institution. In many ways, they sort of operate under the radar. And I just want to take a moment to thank all of you for that, because I think it's really So just another way to look at this kind of gives you a sense of the spectrum of donors that have been involved so far. We have a total of 261 donors that have contributed to this initiative. And I think it really goes to show just the vast array of supporters that we have. And as we look to sort of transition to the next phase of our fundraising, that community support, that broad array of donors is going to be even more important. And so that's really I think our message today is that as we go through this sort of moment in our growth, we really want to reach out to this community to say, we need your help to be stewards of the culture here at MDIBL, to constantly remind us of who we are and ground us as we think as we make some of these decisions going forward. You know, you all really represent MDIBL. You are this community. And so it's such an important time for us to acknowledge that, to move through this process together. And I think we're all really, really excited about what the future holds. So as we continue to think about some of the financial challenges that the lab will continue to face, you know, a big part of this strategic plan is really to strengthen the research programs, to really place the lab on a more secure financial footing. Because as we all know, it's been a repeatable story throughout the lab's history that it's always sort of operated on a shoestring. We want to try to move away from that as much as we can and create some consistency in our funding. Maybe that's a little ambitious, but I think we can do it. And so a big part of that is continuing to strengthen and recruit outstanding scientists that are here. So supporting the folks that are here, recruiting some new folks to the lab, and that will be a big part of the next phase as we implement this strategic plan. So to help us get there, I'm excited to announce that in June, we received a $3.5 million donation. It was a challenge gift. So the donor challenged us to match that amount of money. And already we have been successful. We've raised $1.1 million toward that challenge. And so our goal is to complete this by the end of the year. So we're going to need everybody's help to be able to do that for sure. Emily and I would love to be able to just write that check, but I don't think we're quite there yet. But nonetheless, so we have about $2.5 million to raise by the end of the year. But I think this is a wonderful opportunity for us as a community to really take this next step in our fundraising effort, to really set the stage for what comes next, because we still have a ways to go. We're about 60% of the way to our goal, but now we really want to begin to sort of embrace a broader selection of supporters and take this to the community to be able to succeed. I do want to just take a moment to thank my team, because I think this is an amazing group of folks who are really dedicated to making sure that this is successful. So Emily Burke, I don't know if they're probably out setting tables for lunch, but if we could just take a moment to thank everyone there in the Belton office. And so really, it is just about community. You know, this has been one of those, you know, you're really old when you start achieving milestones in your own life. So Claudine and Chris and I celebrated 25 years of being here at MDIBL this year, hard to believe. But it does make you sort of reflective and think a bit about all that we have accomplished together. And when we were back there in 2001, there were just 10 of us, it seemed pretty daunting that we would ever be in a position to hear amazing science like what Emily presented today, and to now have, as Dr. Haller mentioned, a true research institute that's fully present here year-round. So again, just a huge thanks to all of you for helping make that possible. And I think we're really excited about what the future holds. So thank you all very much.
Well, thank you, Jerry. Thank you, Emily, and thank you, Herman, for those really wonderful presentations. I want to thank everyone that's here today in person and everyone that's here today by Zoom. Without your commitment to the lab, we could not do the work that lies ahead. And your continued involvement, your continued financial support, your continued enthusiasm for this place and bringing it to the attention of others to get them involved and for them to also help realize the future is really critical. So again, I want to thank you very much for being here. I want to remind everybody that you're all invited to lunch in the tent outside. And with that, I would ask for a motion to adjourn the annual meeting. Second?
Second.
All in favor? And no one better object. Thank you so much.